Welcome back to the deep dive. Today, we're really zooming in, not just on a big problem, but on a very specific, highly aggressive enemy within prostate cancer. Our mission, to give you the rundown on a personalized treatment breakthrough. It's aimed, right at the weak spot of some of the most lethal tumor cells out there.
And it's a huge issue globally, about 1.5 million men diagnosed each year.
Exactly. But we're focusing, like you said, on a specific subgroup, one that hasn't really responded well to the standard first-line treatments.
That's right, that specificity is crucial. If you look at the advanced stage, metastatic cancer, the kind that spread well, the picture isn't great. Just in the UK, for instance, you've got over 56,000 men diagnosed every year, and sadly, around 12,000 die from it annually. The research we're digging into today focuses on those patients who, because of a particular genetic flaw, their cancer just takes off. It progresses really aggressively, really quickly, even with standard therapy.
Okay, so let's unpack that. The core problem, the reason this group's cancer is so aggressive, it's down to faulty genetics, right? The cells can't repair themselves properly.
Precisely. We're talking about defective homologous recombination repair genes. HRR genes for short.
HRR genes, okay.
Yeah, think of genes like BRCA1, BRCA2, people might know those from breast cancer,
Right.
but also others like CHK2 or PALB2. They are essentially the cell's high-level DNA maintenance crew. Their job is fixing the big, messy double-strand breaks in DNA. When these genes are faulty, mutated, well, the maintenance crew is broken.
So the damage doesn't get fixed.
Right. And the cancer cells become genetically unstable. That allows them to grow faster, mutate more, and spread much more aggressively.
And this isn't some tiny fraction of patients, is it?
No, not at all. That's the critical part. This kind of HRR mutation, it affects roughly 1 in 4 men with advanced prostate cancer.
1 in 4. Wow.
Yeah, a quarter of that advanced population is dealing with this particularly nasty, hyper-aggressive form. So our mission today is to dive into the phase 3 trial results that offer a really targeted solution, one designed to exploit that specific genetic weakness.
Okay, let's set the scene first. For a patient diagnosed today with advanced metastatic prostate cancer, what's the standard playbook? And why doesn't it work for this HRR group?
So the current first-line treatment usually involves a combination called AAP. That's abiraterone acetate plus prednisone. Abiraterone acetate is a type of hormone therapy, androgen deprivation therapy, technically. It basically works by cutting off the cancer supply of testosterone, which most prostate cancers need to grow.
Okay, so it starves the cancer of hormones.
Exactly. And it's pretty effective for the majority of prostate cancers, the ones that really rely on those hormones.
But you're saying if the cancer cells have this broken HRR repair system, they've kind of got another way to grow, something the hormone therapy doesn't touch.
That's the crucial insight, yeah. AAP targets the hormones, sure. But it does absolutely nothing about the underlying faulty DNA repair system. Our sources show that when a patient has one of these HRR mutations, their cancer tends to progress much, much faster, even on AAP. The HRR defect essentially makes the cancer cells more resourceful, more resistant.
Leading to quicker progression, shorter survival times compared to tumors without this fault.
Exactly.
But if the standard AAP works well for, 3/4 of patients, why change the whole approach just for this subgroup?
Well, because for that 1 in 4, the current approach just isn't good enough. It's not just about treating the disease generically. It's about treating the patient based on their tumor's specific biology. That genetic alteration, usually in BRCA1 or BRCA2, that becomes the critical flag. It tells you who needs a different personalized first-line strategy.
So we need something that doesn't just block hormones, but goes after that fundamental DNA repair weakness.
Precisely. We need to target that vulnerability.
And that strategic targeting. That brings us neatly to the breakthrough trial, the phase 3 amplitude trial. UCL researchers, Professor Gerhard Attard leading it, they added a specific new drug to the standard AAP. What was it? And how does it actually work?
Right, they added Niraparib. Niraparib is what's called a PARP inhibitor.
PARP inhibitor.
Oh, yeah, it's a type of targeted therapy, which is important. It's not like traditional chemo that just hits all fast-growing cells. It's more specific. The goal of the Amplitude trial was simple. See if adding Niraparib to AAP could significantly boost outcomes, specifically for these men with the HRR-mutated cancers.
Okay, we've got the pieces, the broken HRR system in the cancer cells, and this PARP inhibitor drug, Niraparib. But maybe walk us through the sort of aha moment here. Why is blocking PARP so deadly to a cell that already has a broken HRR gene?
Right, that is the absolute key. It's a concept called synthetic lethality. It sounds complex, but the idea is pretty straightforward. Think of DNA repair like having two main safety systems in a cell. The primary one, the sort of gold standard, is HRR, the system run by genes like BRCA.
Okay, the main system.
When BRCA or another HRR gene is mutated in the cancer cell, that primary system is down. It's broken.
So the cell's already vulnerable. Got it.
Exactly. But it's not dead yet. It usually has a backup system it can rely on to patch things up enough to survive. And that backup system uses a protein called PRP.
Ah, okay, so there's a backup.
Yeah. Now, normal healthy cells, they have both HRR and PRP working fine. So if you inhibit PRP in a normal cell, it's usually okay because the main HRR system can still handle repairs. But in these specific cancer cells, the main HRR system is already broken. So if you then come in with the Niraparib and block the PRP backup system...
You take away the last resort.
Exactly. You essentially remove the cell's only remaining way to fix its DNA damage effectively. It's like kicking out the last support beam, holding up a shaky building.
Wow. Okay, so the cancer cell tries to divide, its DNA is already damaged, and now it has no way to fix it. Leads to cell death. That's clever.
It's a very deliberate exploitation of the tumor's built-in flaw. The Amplitude trial was designed to prove this works, in a large group of actual patients. And the scale was decent. 696 men, 32 countries involved. Median age was 68, so pretty representative.
Okay, let's get to the results then, the numbers. For these patients who already have the most aggressive form of the disease, how much difference did this combination actually make?
The results were, pretty striking. The combination Niraparib plus AAP, it did exactly what they hoped. It substantially slowed down the cancer's progression, and it dramatically delayed the cancer coming back.
Can we put a number on that delay? Because substantially slowed sounds good, but specifics really hit home for, the listener.
Absolutely. So for the patients with those critical HRR mutations, adding Niraparib cut the risk of the disease progressing or the patient dying by 40%, compared to just standard AAP and a placebo.
40% risk reduction. That's huge.
It is huge. And the median time until the disease actually progressed, meaning that cancer started growing again on scans, that was significantly longer too. Professor Attard put it plainly. He said the combination can significantly prolong life expectancy just by delaying that recurrence, delaying the pain, the bone problems, all the things that come with progression.
That's not just tweaking things. A 40% risk reduction that basically rewrites the standard of care overnight for these patients, doesn't it?
It really demands that change, yes. The immediate takeaway is crystal clear. These findings make a powerful case for widespread genomic testing right at diagnosis.
So you know from the start who has this HRR fault.
Exactly. If you identify it early, you don't waste precious time on a treatment that's likely to be less effective for them. You go straight to this personalized, synthetic lethal combination.
Right. But shifting gears a bit to the practical side, the clinical reality. These targeted treatments, they're powerful, yes, but they often come with their own set of side effects. There's always that trade-off, that balance of risk versus benefit. What did the safety data from Amplitude show about adding Niraparib?
Yeah, that's the critical nuance, isn't it? You have to look at both sides. While overall the combination was considered generally well tolerated, the data clearly showed that side effects were significantly more common in the group getting Niraparib compared to the placebo group.
More common. Okay. What kind of side effects stood out?
The main things were hematological issues, so blood-related, specifically anemia, which is a drop in red blood cells. That was quite common, and also high blood pressure, hypertension. Those are the most frequent, significant ones.
Anemia. How severe did that get?
Well, this is a statistic that definitely needs careful consideration. 25% of the patients receiving Niraparib needed blood transfusions because the anemia got severe enough.
Wow. 1 in 4 needing transfusions. That's not insignificant. That impacts quality of life, needs extra clinic visits, monitoring.
It absolutely does. It's a real clinical management point.
Yeah.
Even with the clear benefit in slowing the cancer, that level of intervention is a factor.
And what about, the really serious events? Did that differ?
There was a difference there, too, which the researchers reported transparently. Treatment-related deaths, unfortunately, were higher in the Niraparib group, 14 patients compared to 7 in the placebo group.
Okay, that's important context.
It is. Though it's also worth noting the overall rate of patients stopping the treatment entirely due to side effects remained low across the whole trial, which suggests that for most patients and their doctors, the major benefit of delaying disease growth was felt to outweigh the increased risks and the need for closer management of those side effects.
So acknowledging the safety profile, the path forward seems pretty clear based on the efficacy. What did the authors say about next steps? Any key caveats?
Well, they definitely stressed that while this delay in progression is powerful, Right now, more follow-up is needed to confirm the long-term overall survival benefit. We want to see proof that patients actually live longer, not just that the cancer stays dormant longer.
Right, the ultimate measure.
Exactly. And they also suggested future work should look at using newer imaging techniques, maybe monitor the disease even better. And crucially, keep pushing for broader genetic testing so that every patient who could benefit from this approach actually gets identified. This trial really nails the mechanism, but optimizing the real-world execution is the next step.
This has been a fascinating deep dive, really shows the power of genetics meeting oncology. If we boil it down for you, the listener, the key takeaway is this. Advanced prostate cancer treatment is rapidly shifting towards truly personalized medicine. A simple genetic test for these HRR mutations, done right at diagnosis, is looking less like an extra and more like something essential. It could directly tell you the best first-line drug combination, AAP plus that PARP inhibitor, Niraparib. That genetic flaw, which used to just mean a worse prognosis, has actually become the target itself.
That's absolutely right. This research is just a fantastic example of exploiting a known biological weakness. The very thing that makes these cancer cells so aggressive, that defective DNA repair, is precisely what the PLP inhibitor uses against them through synthetic lethality. And it really makes you think, doesn't it? This success in prostate cancer raises a huge question for other cancers. Which other common ones, maybe ones we don't routinely genetically profile right now, might have similar HRR weak spots? Or perhaps other specific genetic vulnerabilities just waiting to be targeted by PRP inhibitors or, similar smart drugs?
An excellent question to ponder as we move deeper into this era of precision cancer therapy. Thank you for joining us on this deep dive. We'll catch you next time.